Good afternoon everyone, I am Salam. Today I will present our recent findings on brain metastasis associated fibroblasts in non-small cell lung cancer and their role in tumor progression and therapeutic resistance. Recently we have published this article. In this article we have isolated CAF from the brain met, and this CAF secreted cytokine IL-26 and CX3CL1 drive the tumor progression through the JAK-STAT3 and AKT-mTOR pathway. Let's see the clinical problem and importance. The epidemiology of brain metastasis could be the different types of primary tumors. Among all the primary tumors, lung cancer account 48%. Among all the lung cancer, non-small cell lung cancer account 26% for the brain metastasis, and the brain metastasis patient will survive 9 to 12 months. Let's see the tumor microenvironment and in the tumor microenvironment CAF are the cell types. These cell types can be detected by those marker expression. In the tumor microenvironment, CAF secreted some factors. These factors can activate the tumor growth, invasion, metastasis, immune modulation, and therapeutic resistance. In tumor microenvironment, CAF secreted some molecule that activated normal fibroblasts to the CAF. This CAF secreted some molecule directly interact with the cancer cell and increase the cancer cell proliferation, stimulus, migration, invasion, metastasis, and therapeutic resistance. Why are we focusing CAF on brain metastasis on non-small cell lung cancer? Because absence of fibroblast cell in normal brain parenchyma and the role of CAF in non-small cell brain metastasis have not been investigated yet. Very earlier, we have published this article related to the differential expression of CAF marker in brain metastasis from the different primary cancer. In the brain metastasis, CAF could be originated from the pericyte, endothelial cell, and astrocyte, and high expression of the CAF marker lead to the poor patient survival. Let's dive to our results section. First, I will discuss about the role of CAF in tumor genesis and therapeutic resistance. First, we have isolated normal fibroblasts from the scalp tissue and CAF from the brain mat, and we have validated our CAF by the expression of CAF marker. Then we have performed the RNA-seq to validate the differentiation of CAF from the normal fibroblast, and those are the differentially expressed gene found in the CAF, and those directly upregulated some pathway like epithelial cell proliferation, and it could be involved in epithelial cell migration and activate the several kinds of pathway activation like JAK-STAT3 and PI3 AKT signaling pathway. Then we have validated the CAF role in the cancer cell proliferation. In the CAF group, when we co-culture with cancer cell, significantly increase the cancer cell invasion, migration, and proliferation, and it also activated the EMT related transcription factors in the cancer cell. So we can say that BM-CAF accelerates the tumor genesis and EMT phenotype of lung cancer cell. Then we have co-injected our CAF cell with a cancer cell, and in our CAF group significantly increased the tumor burden in the mouse. Even in our orthotopic mouse model, we have intracranially injected our CAF cell with cancer cell, and we found that increased the tumor burden in the CAF group compared with the normal fibroblast and control. So we can say BM-CAF co-injected with cancer cell increased the tumor burden. Then we have figured out the therapeutic resistance by CAF. If we look at here, this is the cisplatin treated on cancer cell, 30%. When we added the CAF, it reduced the total apoptotic cell percentage. Even when we added the CAF, the apoptotic protein expression significantly downregulated. So BM-CAF attenuated cisplatin uptake by cancer cell and induced therapy. Then we found our BM-CAF significantly changed the cancer cell phenotype as a stimulus. And so after co-culture, we found the stimulus marker is significantly upregulated in the cancer cell. And further, we have co-injected our CAF cell in the cancer cell with cancer cell and all mice treated with cisplatin, and we have found the tumor burden are increased in CAF group rather than decrease the tumor size. So BM-CAF induces stiffness phenotype of cancer cell and induce therapeutic resistance in vivo. In our co-culture system, we have found the BM-CAF significantly upregulated the phosphorylation of JAK-STAT3 and AKT-mTOR pathway through the parakine. Now move to the result section. Now I will discuss about the mechanism of tumor rejection and therapeutic resistance. Finally, validate our clinical cohort. We have analyzed the KEGG inducement pathway, and in this pathway analysis, we found the cytokine receptor interaction are activated by the DG of CAF, and total 11 cytokine involved on this pathway activation, and among all the 11 cytokines, we have found IL-26 and CX3CL1 are the topmost candidate in our BM-CAF, and even these two cytokines have been found in the BM-CAF secretome also. In our parallel study, we have isolated the fibroblasts from the lung tissue of mice, and then we have activated the normal fibroblast as a CAF-like phenotype by the expression of CAF marker. We have analyzed the secretome of our activated CAF, and interestingly, we have found the CX3CL1 secretory molecule have been found in our activated CAF. So this cytokine CX3CL1 come from the activated CAF and in our BM CAF, and that's why we have selected this cytokine CX3CL1 and IL-26 for our following experiment. Then we have validated these two cytokine related receptor in the all types of lung cancer cell lines. Then we have blocked these two cytokine to see the effect of cancer cell invasion. Significantly decrease the invasiveness of the cancer cell when we block these two cytokine. So neutralizing IL-26 and CX3CL1 attenuated lung cancer cell invasion. Then we have blocked the CX3CL1 to see the pathway activation and significantly reduce the AKT-mTOR and JAK-STAT3 pathway. But in case of IL-26, when we blocked, only JAK-STAT3 pathway have been deactivated. And in our EMT section, we have found after blocking of CX3CL1 and IL-26 significantly reduce the EMT marker expression along with the EMT gene expression. Then we have blocked these two cytokines to see the effective of cisplatin in the cancer cell, and significantly we found when we blocked these two cytokines significantly increased the apoptotic cell percentage. So if you look at here, the total cell percentage 51%, but when we added the CAF reduced the apoptotic cell percentage, but when we block these two cytokines significantly increase the total apoptotic cell percentage. So we can say neutralizing these two cytokines reverse the cisplatin resistance and assist to kill the cancer cell. Then we have validated these two cytokines in our clinical cohort, and this is the high expression of the cytokine and this is the low expression, and high expression of these two cytokine lead to the recurrent free survival and overall survival. That means these two cytokine highly expressed in the brain mat and reduce the patient survival. So this highlight their potential as a prognostic biomarker. So in summary, our findings established the IL-26 and CX3CL1 signaling as a critical mediator of BM-CAF induced tumor progression and therapeutic resistance in non-small cell lung cancer brain metastasis, and this latest clue might open CAF targeted future therapy in non-small cell lung cancer brain metastasis. Now we are investigating the immune modulation by the CAF. So our current project is going on these topics. Finally, I would like to thank my supervisor, collaborators, and lab members for their support and the funding agency for supporting this work.